Welcome to the Deep Dive. Today we're looking at something fundamental in molecular diagnostics, really getting into the difference between the genetic blueprint and the active recipe.
That's right.
Our sources zero in on RNA analysis, why we need it, where it shines, and, well, why sometimes this really fragile molecule is actually our best tool.
Yeah.
Most people, when you say genetics, they think DNA. It's super stable, the long-term archive, the instructions for, well, everything.
The permanent record.
Yeah, exactly. But if DNA is the master blueprint locked away, RNA is more like the work order out on the factory floor, the instructions being used right now. Okay, let's unpack this a bit.
And that right now part is absolutely key. DNA stability is great. It's why DNA testing is often preferred if you're looking for a fixed mutation, something inherited.
Right.
But RNA, that's how DNA actually makes things happen in the cell. If you want a snapshot of function, what genes are actually turned on, being expressed at this very moment.
Whether that's in our own cells or maybe an invading virus.
Precisely. You have to look at RNA, specifically the mature messenger RNA, the mRNA, which carries the codes for all the genes that are currently active.
Okay.
So our deep dive today is really about those situations where, traditional DNA testing hits a wall, maybe technically or just in terms of the information it gives us.
And RNA analysis steps in.
And becomes absolutely critical for certain diagnoses, for managing disease.
Right. And maybe the clearest example is when the thing causing the disease is itself made of RNA. Infectious diseases come to mind.
Exactly. If you're dealing with an RNA virus, well, you analyze its RNA. Makes sense, right?
It does.
But it's not just about detecting it. It's often about quantifying it.
Yeah.
The sources really highlight this for chronic viruses like HIV, human immunodeficiency virus, and hepatitis C.
Okay, quantification. Why is that so important?
Well, think about managing these conditions. A doctor needs more than just yes, the virus is there. They need to know the viral load.
How much virus is actively replicating?
Exactly. How many copies are circulating? That number, that quantitative measure, is basically the gold standard for seeing if a treatment is working.
Ah, I see.
If the drugs are doing their job, that RNA level, the viral load, should drop significantly. So RNA analysis gives you that dynamic, real-time feedback on how well the treatment's working. DNA detection just can't provide that same dynamic picture.
Okay, that makes perfect sense for viral load monitoring. Let's switch gears a bit, though. Cancer. That's fundamentally a disease of DNA, right? Errors in the blueprint.
Generally, yes. Damage to DNA is the root cause.
So it seems a bit weird, maybe counterintuitive, that we rely on unstable RNA here.
Yeah.
But the sources say RNA analysis is key for classifying tumors, sometimes better than DNA alone.
It does seem counterintuitive at first glance, but this is where we get into something called mRNA expression profiling.
Okay.
See, cancer isn't just cells with static DNA mistakes. It's a disease of deregulated activity, finding a mutation in the DNA. That's step one. Step two is understanding which genes that tumor is actually turning on or turning off because of that mutation, or maybe other factors. What's it actually doing?
So the activity profile.
Exactly. That expression profile gives you a much clearer picture of the tumor's current behavior, its potential aggressiveness. It's become a really important tool for molecular stratification.
Stratification, meaning grouping them.
Yeah, classifying tumors into subtypes that behave differently or respond differently to treatment. The sources mention breast cancer specifically here.
Okay.
This kind of profiling can help predict, say, the risk of recurrence or identify active pathways that you could potentially target with specific drugs. It lets doctors look beyond just, where the tumor is and what it looks like under a microscope.
And understand its unique molecular engine. Sort of.
That's a good way to put it. It's active machinery.
Fascinating. So we go from finding a static error to analyzing dynamic behavior, which brings us to what I found, frankly, the most surprising point in the sources. The technical reason why RNA can sometimes be way, way more sensitive than DNA for finding specific genetic changes in cancer. Here's where it gets really interesting.
Right. This is a neat biological trick, actually. It hinges on how cells process their own genetic information. We're talking specifically about chromosomal translocations.
Okay, translocations. That's when bits of chromosomes break off and stick together incorrectly.
Exactly. Big genetic accidents. A piece of one chromosome breaks and fuses onto another one. This often creates a brand new fusion gene that can drive the cancer. Certain leukemias are classic examples.
Got it.
Now, in the DNA itself, these breaks, these fusion points, often happen within these huge, sprawling regions of non-coding DNA. They're called introns, massive stretches that don't actually code for protein.
Okay, so the breakpoints are hidden in these large non-coding areas.
Hidden and often very far apart. Now imagine trying to detect that specific abnormal connection using DNA PCR. PCR works by amplifying small, targeted bits of DNA.
Right, like finding a needle in a haystack.
Worse, it's like trying to confirm a specific connection point between two ends of a broken string that are lying somewhere across, say, two football fields, using just a tiny spotlight. Designing a reliable DNA test to pinpoint those breaks scattered across tens of thousands of DNA bases. It's incredibly complicated. Often, it's just not practical. But the cell itself does some editing. When it makes the mature mRNA copy from that messy DNA template, it performs a crucial step called splicing.
Splicing, that removes the introns. Right?
Exactly. It cuts out all those massive intervening non-coding introns and stitches the coding bits, the exons, together.
So wait, the cell basically cleans up the evidence. It takes this huge, messy break in the DNA.
And through splicing, it creates a clean, compact, continuous fusion transcript, the resulting mRNA product.
Ah. So the abnormal connection that was spread out over maybe thousands of DNA bases is now right next to each other in the RNA.
Precisely. You get a single, neat, easily detectable mRNA sequence that contains the fusion. It's much, much shorter and highly concentrated compared to the sprawling DNA break.
That's clever.
It is. And that resulting compact sequence is exponentially easier to find and quantify using RNA-based methods than trying to find the original break in the DNA. This whole splicing process is why RNA analysis is often superior for detecting these specific gene fusions.
And the classic clinical example they give is the BCR-ABL fusion transcript. That's the hallmark of chronic myeloid leukemia, CML.
That's the textbook case. BCR-ABL drives CML. Detecting that fusion RNA is key.
Okay, but that raises a practical question. If RNA is so useful here because it's this process compact message, it's also notoriously fragile, isn't it? Doesn't that make working with it in the lab difficult? More expensive, maybe.
That's a very important practical point. Yes, RNA is unstable. It degrades really easily. You have to handle samples very carefully, use special stabilizers.
So how do labs get around that fragility to actually do the analysis?
They use another clever biological tool, an enzyme called reverse transcriptase.
Reverse transcriptase, that sounds familiar. From retroviruses,
Yeah.
like HIV.
Exactly. It's the enzyme viruses like HIV use to copy their RNA genome into DNA. Labs borrowed that trick. We add reverse transcriptase to the patient's RNA sample.
And it makes a DNA copy.
It makes a complementary DNA copy, or cDNA, from the RNA template. You can think of the cDNA as like a stable, durable photograph of that fleeting RNA message.
Okay, so you convert the fragile RNA into more stable cDNA.
Right. And once you have that stable cDNA, you can then use standard, powerful techniques like real-time PCR to analyze it reliably and quantitatively.
So the whole process is reverse transcriptase PCR, or RT-PCR.
Correct. RT-PCR.
And this RT-PCR isn't just for the initial diagnosis of CML, is it? The sources stress its use for monitoring minimal residual disease. That sounds incredibly sensitive.
It is. Monitoring minimal residual disease, MRD, is arguably one of its most powerful applications. After treatment for CML, a patient might look like they're in complete remission by standard tests.
Right. No obvious signs of cancer.
But this RT-PCR method is so sensitive, it can detect maybe just a handful of remaining cancer cells still carrying that BCR-ABL fusion RNA signature.
Wow.
So clinicians can track the level of those residual cells over time, see if it's rising or falling, and potentially adjust treatment long before the patient actually relapses clinically. It's a huge advantage for managing the disease long term.
Okay, that's incredibly powerful for these established uses. Let's look ahead. The sources also suggest RNA analysis itself is sort of on the verge of a transformation, using newer high-throughput methods.
Yes, the field is definitely moving forward. We're looking towards the impact of next-generation sequencing, or NGS.
NGS, that's usually associated with sequencing whole DNA genomes, right?
It is famous for DNA sequencing, but you can absolutely apply NGS technology to RNA as well. When you do that, it's called RNA-seq. Basically sequencing the entire transcriptome, all the RNA molecules in a sample.
Okay, RNA-seq.
And the source material notes that this is becoming, well, surprisingly straightforward to do, and it makes a bold claim that RNA-seq could, in principle, eventually replace almost all of the targeted genetic tests we use today.
Including the RT-PCR methods we just talked about.
Including those current RNA analysis methods, yes.
Whoa. Replace almost all current tests. That's huge. What's the practical difference for patient care between the targeted RT-PCR for BCR-ABL and this broader RNA-seq approach.
Think of it as the difference between fishing with a specific lure versus surveying the entire lake.
Okay.
The current RT-PCR for CML is highly sensitive, but it's targeted. We're only looking for that one specific thing.
The DCR-ABL fusion transcript. It's like fishing just for trout, right?
RNA-seq, on the other hand, is like doing a comprehensive ecological survey of the entire lake. It doesn't just look for one specific RNA. It attempts to sequence every active RNA molecule in that sample all at once.
So you get a global picture, everything that's being expressed.
Exactly. A high-resolution, complete map of all the active gene expression in that cell or tissue sample at that moment. And as the cost of NGS technology keeps dropping,
Which it has been, dramatically.
right, it's pretty reasonable to expect RNA-seq to become much more common in routine diagnostics.
Meaning faster, maybe cheaper in the long run, and definitely more comprehensive information.
Potentially, yes. You'd get a much richer picture of everything that tumor cell is doing, not just the one fusion gene you decided to look for beforehand.
So let's try and tie this all together. We started contrasting DNA as the stable blueprint and RNA as the active recipe. And the core takeaway seems to be that RNA isn't just some messenger boy. It's a really critical dynamic indicator. It tells us what the cell is actually doing functionally right now.
That really is the crux of it. So what does this all mean for you? Well, RNA analysis gives us that vital dynamic snapshot in essentially 3 key areas we discussed.
Okay, to recap.
1, monitoring viral load for infections like HIV and Hep C, measuring the active replication. 2, molecular stratification of tumors, classifying cancers based on their active gene expression profile, like in breast cancer.
Right.
And 3, providing that super sensitive way to detect specific gene fusions like BCR, ABL, and CML, because the cell's own splicing machinery basically highlights the abnormality for us.
Makes it easy to find.
It's much easier, yeah.
So we've seen RNA gives us the dynamic activity, not the static structure. And that leads to a final thought. If we can routinely use this dynamic snapshot from RNA, what are the possibilities? Think about this. What if we could use RNA analysis not just to monitor disease that's already established, but to catch the very first functional signs? The earliest shifts in gene activity when cells start going slightly wrong.
Before a permanent DNA mutation even locks in.
Exactly. Before the structural damage happens. Could we detect disease risk or onset purely based on functional changes in RNA expression? Something for you to chew on until our next deep dive.